As early as December 2019, the new coronavirus began to spread. On March 11, 2020, the World Health Organization announced that COVID-19 was a pandemic. Researchers and pharmacologists raced to get drugs, treatments, and vaccines available for patients and the general public. We interviewed some experts on this topic of drug development.
My name is Arash Naeim. I am a professor in hematology, oncology, geriatrics, and in bioengineering, and I'm the chief medical officer for clinical research for UCLA Health. Clinical trials, by definition, encompass anything that is done in humans that involves either a drug or device or an intervention. And so it encompasses everything from phase 1, 2, and 3 clinical trials. Phase 1 trials usually are looking at the safety. Phase 2 trials extend the safety testing, but also are looking for whether there is a signal that shows that there may be a benefit of the drug in a certain population. And then phase 3 studies compare whatever the clinical trial drug is against whatever the standard is. It could be placebo or nothing, but if there is already a standard of treatment out that is being used, it would compare it against them, because at the end of the day, the FDA is looking for things that are improvements upon what we're currently doing for a particular patient population. In terms of how long it takes for the whole clinical trial activity to happen, typical drug development takes years and years, and a good chunk of that is the preclinical. Then, of course, it does take a little bit of time to do the phase 1 and 2 clinical trials, but oftentimes it's the phase 3 clinical trials that require much larger populations of patients in a randomized fashion with good follow-up, so the FDA feels comfortable making some treatment available to the general public. Trying to develop a new drug from scratch is not a realistic way of trying to do something immediately for people that are sick in the hospital, because that takes time.
As Dr. Naeim says, it's not efficient to have to make drugs from scratch during an emergency. Luckily, Dr. Damoiseaux, the director of the MSSR at UCLA, has a solution.
My name is Robert Damoiseaux. I'm the director of the Molecular Screening Shared Resource here at UCLA. And I'm also professor of the Department of Molecular and Medical Pharmacology, and also professor of the Department of Bioengineering. So Molecular Screening Shared Resource is a drug discovery laboratory, and we develop new drugs. Repurposing drugs is actually a very, very effective and elegant way of finding new cures for diseases that don't have a lot of either resources attached to them or time. You look, for example, in drug libraries of drugs that are already approved for a certain indication. And very frequently, you find that drugs that work for one disease also have an involvement in other diseases. And what this does for you is basically it cuts a lot of time and effort and money that you have to spend otherwise in getting a drug from your bench to the bedside.
I'm Vaithi Arumugaswami. I'm an associate professor in Department of Molecular and Medical Pharmacology, UCLA. So Robert, I know Robert in the last 15 years, when this COVID-19 pandemic started. I think it is mid-March. I reached out to him and I said, this outbreak become worse, and whether we can, join hands to identify some drug compounds. Within a week, we tested close to 435 compounds, identified 35 compounds from that list, and we finally selected 1 or 2 compounds to move further. So it was very productive collaboration for a short period of time we put together. We could actually target about 4 or 5 steps in the virus replication cycle. If we just target one cycle or one step, the virus can become resistant. They mutate and become resistant to that particular drug. So it is important if we can target multiple steps of the virus replication, so we could actually completely inhibit the virus growth. We could also target the cellular or the host factors that is or that are critical for viral replication. So we could actually have parallel strategies. One that you could target the virus, the other one you could target the host factors. So in that way, you have complementing approaches to completely, stop this virus and make treatment options to cure this disease. When you are targeting a host factor, the host factor may be crucial in doing certain function in the host cell. Sometimes you get adverse reaction. So, however, if you can actually zoom in to the exact interaction of the host protein with viral protein, and if you could specifically disrupt that interaction, it could be physical interaction or functional interaction, you can actually minimize that side effect. So that's why the drug development, it goes through a series of steps. So we will have multiple gates to find out if the drug is safe for that particular ailment without causing adverse reaction.